Hi, and welcome back to another episode for this biology series. So today we're going to follow on and start talking about cells. And I'm going to introduce you to probably the most simple, direct cell design that you can possibly think of. And that is this little fella here. This is what we call a prokaryote cell. And they're usually a type of bacteria. So they're very simple in cell design and very, very small in size. They're only around about the 0.2 to 2.0 micrometers. So the little U in front of the M there, it stands for micro because mm is already taken up by millimeters, so you've got micrometers with the UM. They've also got no membrane-bound organelles, and an organelle is a compartment or structure inside a cell. And these ones, they do have some structures. As you can see, they have a very simple structure. Bacteria, and we'll go through that in a minute, but none of them are bound by a membrane except for the prokaryote or the bacterial cell itself. They also don't have DNA that is structured the same as ours in linear chromosomes. They have a circular DNA. They do have a chromosome structure, but it's just a circular DNA strand. They have a cell wall as well, much like our plant cells do that you've probably heard of before, but their cell wall is complex. It's a combination of proteins and carbohydrates called a peptidoglycan cell wall. So we're going to run through some of the structures that exist on this cell, and if I was just to direct you to this area here, this is where the cell wall would be, and just underneath it would also be the cell membrane. So that cell membrane we'll talk about in later lessons, but it's an important one. It's made up of a special kind of structure called a phospholipid bilayer. Other things to note is that they do in fact have structures on the inside of them, these ones being our ribosome structures. And this large circle here is where you would find your circular DNA. So other things to notice that they're generally covered in moving or structures as well, things that allow the bacteria to move. These little ones here are what we call cilia. They're like tiny little finger-like projections, hairs that will allow the cell to move around. But the main big bad that allows things to move around is this large tail that we call a flagella or flagellum. Cells can have multiple flagellum. This little basic one that I've gone here is just just a one flagellum, just to show how the structure of it looks. And it's all sitting inside of a fluid-like membrane. This one here would have a cytoplasm as well. Yeah, there we go, it's going to fit. Perfect. Another thing to note is that a lot of bacterial cells also have a capsule that surrounds the outside of them, and it's kind of like another layer of protection that allows it to be able to slip around and invade other cells or other organisms and go undetected, but also adds a protective layer for the cell. As a lot of the time, these prokaryotic cells, they exist as a single-celled organism. They're not multicellular, and they're just a one cell. Sometimes they live in colonies. Most of the time, they're just individual cells that are able to operate all by themselves, operate all on their lonesome. So their size is in fact important, and I'm going to be talking a little bit about that in a moment, but just to compare our dear friend, the prokaryote cell here, to its larger friend. I mentioned its size before. If we were to shrink it down, that 0.2 to 2 micrometers, there's our big bad boy that we always know and love, the one that always gets spoken about. This is our example of a eukaryote cell. So we know these cells, or you should know these cells. They're used as the majority of the example. And this is just your standardized cell that I've got here as a bit of an example. They're also a small size, but they're larger than the prokaryote. They exist in the 10 to 100 micrometers zone. We can even see some of those larger cells, not quite with the naked eye, but if you're looking carefully enough, if you think to yourselves, can you see yourselves, the individual skin cells on, say, an onion or something like that, under a low power magnification. These guys do have membrane-bound organelles, these compartments on the inside of the cell. And you can see here that that the organelles are all organized. I've got them, color-coded and fancy, drawn up and things like that. And we'll run through the individual roles and functions in a later episode, but for now we just need to know that they exist, and we're going to talk about why they exist inside of our larger cells to begin with. We have ourselves linear DNA existing as chromosomes inside the nucleus. And if there is a cell wall on a eukaryotic cell, it is a simple cell wall. If you think about a plant cell, it's only made up of cellulose. So it's a very simple plant cell wall compared to that of the prokaryote cell. And there it is, our membrane-bound nucleus. So I could run through some of these important organelles going on inside our eukaryotic cell. You've got the nucleus over here, and the accompanying nucleolus. That's really important, as the word eukaryote means good or true nucleus, while prokaryote means before nucleus. So this guy here exists. This is our control center. It's where all the DNA is stored and kept inside of our cells. We have ourselves a mitochondria. And if you look, the mitochondria and the prokaryote cell are roughly the same size. There is a theory that mitochondria actually are descended from prokaryotic cells, and they help us with energy production. Here is our smooth ER, our smooth endoplasmic reticulum. Here's our rough ER over here. And again, floating inside are our ribosomes. Now, they would be similar in size to the ribosomes that would be found in the prokaryotic cell. I've just drawn them a bit bigger here so that I can see them. And our lysosomes as well. Oops. Let's undo that. There we go, lysosomes, and our Golgi. Now, this would be an example of an animal cell. You can generally tell that from the shape, and on the outside of an animal cell, there is no cell wall, but there is a cell membrane. And it would have a very similar, if not exactly the same type of membrane structure as to the prokaryotic cell. So we do have ourselves a lovely, example of what the plant of a eukaryotic cell would be. So the big question to go on here, the big question to go on with here, sorry about that, is, well, why exactly do we have organelles anyway? Why would the prokaryotic cell have no organelles, while eukaryotic cells, which is what human beings are made up of, human beings, other animals, and plants as well, do in fact possess? Oh, why organelles? Well, I'm going to throw out this question here and do the one thing that a lot of people hate, and that is put in some maths inside biology. So the maths here has to do with surface area to volume ratio. I'm going to pose this quick question here. Can you do the surface area to volume ratio of these two cubes? If I was to tell you what the measurements are, I'm going to tell you that this here is a 1 centimeter cube, and this one here is a 3 centimeter cube. Now, if this is a lesson, you can pause the video here, quickly do the math, see how you go, and then we'll compare it. But I'll give you a second to decide to pause or not. Otherwise, the answer is quite simple. If you were going to do surface area to volume ratios for each of them, well, you're going to calculate the total surface area of this cube, the total surface area of this cube, and then compare it to the total volume of this and the total volume of this. Surface area is simple enough for a cube. Because cubes are all squares, making up the panels, we do length by width of a square. In this case, it's going to be 1 times 1. And then you multiply it by how many panels that we have. So that's a total of 6. And so our surface area here is going to be 6. Volume, again, is very simple for a cube. Length times width times height. So this is going to be 1 times 1 times 1. And you end up with a 1 for the volume as well. The 3 by 3 by 3 cube, similar situation. Surface area equal to 3 by 3 by 6. So essentially that's going to be 9 times 6, so you got yourself 54. And then volume is equal to 3 by 3 by 3 for length times width by height. And that is a grand total of 27. So now we compare our surface area to volume ratios. This one would have a, whoopsie, I think undo. Okay. Surface area to volume ratio will be a ratio of 6 to 1, and that is as simple as it gets. A lot of people will just say it has an SA to V of 6 though, because they count as this as a fraction, so 6 divided by 1 will just be equal to 6. While our larger cube, which has much more space on the outside, but also much more space on the inside, has a surface area to volume ratio of 54. Whoops, 54 to 27. And you're probably thinking, great, excellent, that large cube has a much greater surface area to volume ratio, which would be true until you simplify it down so that they're about the same, in which case you end up with a ratio of only 2 to 1, which is a division of 2. So the larger an object is, the smaller its surface area to volume ratio. This is important for cells for the purpose of efficiency. So you want to have more surface in contact with the outside environment so that the cell can continue to exchange with that environment. Some of you may be aware of an experiment that you can do that involves agar cubes, sodium hydroxide, hydrochloric acid, and some kind of indicator. So you can set it up very simply, get 3 cubes that have 3 cubes of agar, one that's 3 by 3 by 3, 2 by 2 by 2, 1 by 1 by 1, or whatever sizes you can manage to make. And in the agar, you can dissolve or prepare it with some sodium hydroxide. That will make it basic. Give it an indicator that allows it to detect when basic turns into acidic, or when basic turns into a neutral reaction, I should say, and you can actually see this reaction in state. So in this particular instance, we use 1 molar hydrochloric acid, and we use 0.4 molar hydrochloric acid and 1 molar sodium hydroxide. This allows the reaction to happen a little bit more slowly, and so you can see it over time instead of just seeing it immediately. Either way, this is the reaction. So you can set it up with the agar cubes completely submerged in the hydrochloric acid solutions, and then eventually what happens is the hydrochloric acid permeates into the agar, which is something that is meant to happen. It doesn't dissolve, it doesn't burn it away, it dissolves in there, and as it dissolves in there, it neutralizes the basic inside of the cube, allowing there to be this color change. When the color, you can then take these cubes out and then see what percentage of the cube has in fact been changed color of. And this will give you an idea of how surface area to volume ratio affects the efficiency of cells. Cells want to be able to exchange with their environment. They want to be able to get rid of waste and take in nutrients quickly and efficiently. And having small packages or being of smaller size with much more surface area in contact with the outside world is of benefit. So what does that have to do with our prokaryote and eukaryote cells in the end? Well, it also allows, smaller size also allows for more reactions to take place. So if you have so many molecules inside of a cell, we want our molecules that need to react together to find each other, to collide, and then cause our reaction. Now, this can happen in cells of medium size quite easily, and it can also happen much easier in smaller cells, which means that prokaryotes are in fact of advantage here. They're able to exchange with their environment very, very quickly, and they're able to get a lot of reactions happening inside of them very, very quickly. Simple design, small size, lots of efficiency. When we increase the size of a cell, however, it can be really easy for our molecules to miss each other completely. And random reactions can take lots and lots of time to actually take place. So eukaryotes came up with this way to not only increase surface area to volume ratio by giving themselves more membrane space, hence membrane-bound organelles, but also to give themselves smaller compartments for reactions to specifically take place inside of. So here's our eukaryotic cell, and here are our compartments, its organelles. Suddenly, the reactions can take place a lot easier, the molecules find each other a lot easier, and it becomes a lot more efficient. Exchange becomes a lot more efficient. Reactions become a lot more efficient. Eukaryotic cells and eukaryotic organisms can increase the amount of surface area to volume ratio that they have by increasing the amount of surface area each cell has, decreasing its volume, but changing the sizes, such as the villi cells inside of your intestines and microvilli inside of your lungs. The whole idea is that these guys increase the surface area that they're in contact with and limit the amount of volume that they have to increase that exchange that is happening. So the microvilli are in contact with lots of blood and lots of oxygen on either side. They're in contact with a little bit of blood on the inside, lots of oxygen on the outside, and they're able to exchange that gas, carbon dioxide out, oxygen in, really, really efficiently. And the same goes for your villi inside your small intestine, absorbing nutrients back in and letting go of waste product nutrients out. So we're up to the lesson recap time. We've gone over that simple cell design being the prokaryote. We looked at prokaryote versus eukaryote, the importance of cell size and surface area to volume ratio, and how exactly organelles and cell shape can increase surface area to volume ratio. I hope you found this informative, and I look forward to putting up the next episode soon.